So it's my pleasure now to introduce a longtime friend and colleague, Professor Linda Penn. Linda is a senior scientist at the Princess Margaret Cancer Center and a professor in the Department of Medical Biophysics at the University of Toronto. She's also the inaugural director of the University Health Network's Office of Research Trainees, so certainly is committed to supporting research trainees. She is a scientific advisor on several national and international review panels and is on several journal editorial boards, including senior editor of Cancer Research. She's been honored with many awards, the most recent being an honorary degree, doctorate degree from Linköping University in Sweden. Dr. Penn undertook her PhD studies at the University of Toronto at the Hospital for Sick Children with Dr. Brian Williams. And at that time, she focused on interferon as an antiviral, hence our long-term friendship, because as you all know, I'm absolutely committed to interferons. She then pursued her postdoctoral fellowship in the UK at the Imperial Cancer Research Fund in London with Drs. Helmut Land and Gerard Evan to study the MYC oncogene in cancer. And that is where her journey has continued both in the UK and in Canada. So moving back to Canada, she established her lab with a very specific focus on the regulation and function of MYC in cancer. And most recently, she's added another interest in the context of statins. But this morning, Dr. Penn, or this afternoon, this evening, wherever you are, Dr. Penn is going to tell us about how to drug the undruggable c-MYC. So Linda, over to you, and looking forward to a great presentation. Thank you.
Thank you. Thank you all. In fact, I must start by commending you. Beyond Science's initiative, very inspiring work. You were way ahead of your time, making use of virtual communication for outreach and collaboration, way before the rest of us had to learn, basically, that this new medium basically enables this global outreach. So congratulations. I'm very honored to be here. Thank you. All right, let me start to share screen. Can you see that?
Yes, we can.
Okay. I have to get rid of you, Eleanor. You're in my way. All right. So as Eleanor mentioned, today we'll be focusing on how to inhibit the undruggable MYC oncoprotein in cancer. And here I just show a picture of Toronto. So when we can all travel again, I hope that many of you can visit us in Toronto. It's remarkable. It's like a hub of research. There's 8 research institutes in one city block. And whether you can visit us in person or virtually, this is sort of our environment. And that's where, of course, I have been working alongside with Dr. Fish for years. All right, let's focus on MYC. So just to bring everybody up to speed, just to remind you that MYC basically is a nuclear phosphoprotein that basically is associated with cell growth and proliferation. And so in normal cells, MYC is very, very highly regulated. You don't want to have an oncogene like MYC out of control, because of course that will lead to cancer. So MYC generally is a 30-minute half-life protein, very highly regulated, indicated here downstream of all sorts of different signal transduction pathways. And so being such a short half-life protein and connected to all these signaling pathways, MYC functions like the intracellular sentinel of the extracellular milieu. It's really keeping tabs on what's going on in the local environment of the cell, and then being able to very quickly integrate and respond accordingly, enabling that normal cell to grow when safe and to not grow when the environment would not support cell growth. Now, in cancer, by contrast, MYC is, of course, dysregulated in many cancers. And usually, pathologists score a MYC deregulated allele by its gene amplification. But that's not the only mechanism of deregulation of MYC. if any of these signaling pathways, for example, let's say RAS is deregulated, then, and my slides aren't advancing. That's too bad. Oh, don't know what happened. Oh, there. Okay, so if RAS, for example, is deregulated, we would have an activation of this classic ERK-MAP kinase pathway, and that would lead to MYC deregulation. Similarly, if we inhibit any of the anti-proliferatives, so for example, the loss of a tumor suppressor, again, MYC becomes dysregulated. So when we say that MYC is deregulated in over 50% of human cancers, this is why. So if we take any one cancer, let's say breast cancer, classically MYC amplification would be scored at about 15% of all cancers. But if we also consider that PI3 kinase pathway is activated in most human breast cancers, in fact, that also leads to MYC deregulation, as does activation of EGFR families such as HER2, or inactivation of tumor suppressors such as PTEN and SMAD. We were talking earlier about pancreatic cancer, for example. RAS is a classic activated oncogene, and RAS has been shown to stabilize MYC. So it's no longer turning over every 30 minutes, but now is being expressed at a time when it shouldn't be, and able to then act as an oncogenic driver of cancer. Okay, what does MYC do? MYC is a transcription factor with its obligate partner MAX, basically can activate or repress gene transcription. The remarkable piece is that it's actually doing that on 10 to 15% of all genes in the genome. And that's why it has such a broad reach and can regulate so many biological activities. Inhibit differentiation, keep the cell in cycle, induce genomic instability, angiogenesis, migration, growth, proliferation. And I know everybody here is focused on immune response. MYC has been shown to suppress the immune response of the cancer. And so basically, again, broad reach. So many people have said over the years, well, if we could inhibit MYC, that would have a remarkable response. We'd be, have a great advance. But the concern is every cell in a normal body, every cell in your body is expressing MYC, and it's part of normal biology. So how would we ever be able to drug MYC and yet retain a tumor normal index, making sure only the cancer is affected and not all cells of the body? Well, some wonderful proof of concept experiments have been conducted that shows that if we could ever develop an inhibitor to MYC, we could indeed eradicate cancer. So I'm going to show you one example, but there's lots. So here, for example, like I showed you, MYC and MAX drive all sorts of biological activities. A dominant negative sort of research tool was developed. It's called OMOMYC. And basically, as shown here, OMOMYC can displace MYC and MAX from binding to DNA. It's just a little piece of protein, about 92 amino acids, that basically has a few mutations in it, and so acts as a dominant negative to displace. Nothing that could ever be used as an inhibitor necessarily in humans, but a wonderful research tool. And what Laura Susek did when she was in Gerard Evans' lab is she made a transgenic mouse with OMOMYC under the regulation of a doxycycline-inducible promoter. Okay, so it's silent in the mouse until activated. Okay, and it would be activation would be systemic, so both in normal and cancer. So she was in a position to be able to ask the question, if I inhibit MYC in the whole body of a mouse, what happens? And so she did that in the context of the Tyler Jacks RAS-driven RAS-driven lung cancer, which really mimics the human disease. And without treatment, the mice succumb to their disease, here measuring survival. But what she did is she turned OMOMYC on with doxycycline, 3 or 4 weeks, and then much like a chemotherapeutic approach, then turned off. So gave this cycling of basically inhibitor and then a drug holiday, if you will, or an OMOMYC holiday, and then back on again, and then off again. So the normal cells basically, when they're met with an inhibition of, for example, MYC, they still have the capacity to exit the cell cycle and just sort of wait it out. Whereas the tumor cells are so addicted to MYC. MYC is driving that cancer. And without it, basically, they succumb. And so as you can see with this OMOMYC on-off treatment regime, basically the mice lived a long and normal life. A tumor was completely eradicated. And similar experiments have been done by Dean Felcher and others using switchable MYC alleles. And it doesn't matter what the cancer type. He interrogated many different cancer types. Basically, as soon as he turned MYC off, the cancer underwent apoptosis or underwent differentiation. Each cancer type had its own sort of exit strategy once the MYC was inhibited, but the result was the same. Basically, tumor eradication, but the normal cells were fine. So this was great. I was very excited about this series of experiments because it provided people like me sort of the justification or proof of concept that if we could drug MYC, if we could inhibit MYC, we could have a major impact on all sorts of cancers, over 50%. Only one problem. Targeting MYC would have a significant advance, but it's not possible. So unfortunately, MYC does not have the structure that is consistent with classic drug development. So, for example, a kinase has a nice deep pocket where a small molecule can block basically ATP interaction with the kinase and block kinase activity. By contrast, MYC is like a spaghetti. It's intrinsically disordered. It basically has no shape of its own. And so that's why drugging MYC has just been not possible using classic drug development strategies. However, studying intrinsically disordered proteins, we've come to learn that when MYC interacts with when an intrinsically disordered protein interacts with its partner protein, it takes on the shape of its partner protein. And different partners, different shapes. So that said to me, well, then if we could inhibit MYC from interacting with those partner proteins that are very important for MYC to be able to function as a potent oncogene, then we should be able to disrupt MYC activity. Well, that's a very nice hypothesis. So the idea is that basically MYC interacts with proteins in order to drive oncogenesis, and if we can inhibit MYC protein interaction, then we can block transformation. And so there goes the motto or the mission of my lab. That's very nice. However, we really didn't know very many MYC interacting proteins because MYC is highly labile. I told you 30-minute half-life. You just start to break open the cell and MYC just falls apart, let alone MYC and its interactors staying intact. So identifying MYC partner proteins using traditional biochemical approaches was just not possible. So we had to take a new strategy, and we did so. And if you take nothing else home from this talk, please learn about MYC BioID or BioID that you can apply to your protein of interest, because it allowed us to basically identify the MYC protein interactome for the first time. And it's such a clever little assay. And we did this in collaboration with Dr. Brian Rout, who's a mass spec expert. But basically, you make a fusion protein of your protein of interest, in my case, MYC, with BurA. BurA is a biotin ligase. And so wherever MYC goes in its path, within a cell or within a tumor, we actually introduced this fusion protein into cells and then introduced those cells and established a tumor in a mouse. And as long as we provided exogenous biotin, basically the biotin ligase then deposits a biotin on any neighboring proteins. So wherever MYC is going in its day-to-day life, all its neighbors would become biotinylated. Well, as many of you know, then we could conduct harsh lysis, streptavidin pull down, and conduct mass spec. And so only those proteins that were biotinylated basically were identified. And only proteins that are biotinylated are MYC interactors. There's very few endogenous cellular proteins that are biotinylated. Most of those, there's about a handful, are known and have to do with mitochondrial activity. In fact, they serve often as a positive control for us. And so with that, we were able to identify the MYC interactome, like I said, for the first time, and we have hundreds of interactors. Now, this includes direct and indirect interactors. So it's really proteins within the vicinity. We think it's, back of an envelope calculation, we think it's 1, 2, maybe 3 proteins deep in terms of which complexes would be captured. We identified MAX, of course, the obligate partner, as well as known interactors such as TRAP, shown here in yellow, and its associated histone acetyltransferases. So that gave us confidence that this approach really has merit. So today I'm going to tell you a story about one of the interactors we've since studied in more detail, but of course we've got many on the go, as you can imagine. It's been a treasure trove of interactors that we can now study. So one of the interactors that we identified with BioID was PP1 PNUTS. So PP1 is protein phosphatase 1, and it's made up of catalytic subunits, alpha, beta, or gamma. And this phosphatase is basically brought to its substrates by numerous different regulatory subunits. In this case, we identified PNUTS, and that makes sense because it's the regulatory subunit most often used by PP1 to identify substrates, particularly in the nucleus. And of course, MYC is a nuclear phosphoprotein. And that was the other reason we looked to the PP1 PNUTS to further study, because we and others had already shown that MYC phosphorylation is very important to MYC activity. MYC phosphorylation contributes to turnover. MYC phosphorylation contributes to different activities of MYC and can be regulated both pro-oncogenic and anti-oncogenic through phosphorylation. So when we identified this enzymatic complex, and enzymatic complexes themselves could be targetable, we had to follow through. And just to give you an idea of what the data looks like, this is the kind of data you would get. I'm showing you because I'm trying to encourage you to use BioID in your research if you can. But if we use just a Bure control in cells compared to Bure MYC, and then we conduct that strep-david and pull-down in mass spec, you can see that here we have two different biological replicates, each with two technical replicates. First, we see that we pull down MYC. That makes sense compared to control. We also were able to pull down peptides associated with MAX and other sort of known interactors like TRAP I was telling you about. And when you compare the peptides that are pulled down with flag MYC compared to flag control, then you can basically analyze the enrichment and come up with what we call a Saint score. And anything that's greater than 0.8 really means it's likely an interactor with MYC. And so when we saw, this is the PP1 PNUTS data, here's PP1R10 is actually PNUTS, scored well in terms of Saint score, as well as two different subunits of PP1, alpha and gamma. Again, very high scoring interactors based on BioID. And so that proposed this model that MYC actually interacts with PNUTS and PP1 as part of the regulatory subunit. Okay, so first we had to, with all our interactors, we have a series of criteria of what is sort of a go, no-go decision on which ones to further pursue. So our number one criterion is that basically PP1 PNUTS have to also be dysregulated in cancer. Otherwise, we can't inhibit MYC and its interactor if they're not both dysregulated in cancer. We want to make sure that they're both expressed and functional in cancer. And we were able to do that by analyzing C bioparticle. And the beginning of this story is published. It's the latter half that is brand new. So if you want to understand more about this story, you can always look up the paper, which I'll give you a reference for in a minute. Okay, so first order of business is to note that both were dysregulated in primary human cancers. Second, we needed to validate, of course, the interaction, and we do that by a number of different assays. Here's shown proximity ligation assay, and I really like this way to validate a protein-protein interaction within cells because it's very similar to what we did with BioID. It's an in situ labeling, right? So we're not, again, we don't need to break open the cell in order to measure this interaction. Breaking open the cell could disrupt the interaction and therefore lead to a false negative. And so by this approach, we can evaluate within cells whether these proteins of interest are in the same vicinity. So basically, we have an antibody to MYC alone. You can see there's very little labeling. And here you can see an expansion of this one little block. Very few cells are labeled using this PLA. And I'll tell you about that in a minute. PNUTS alone, we get very little signal. But when we assay for MYC and PNUTS using two different antibodies, basically if those antibodies are in close proximity, then basically we can amplify that signal through a rolling circle amplification and actually see plenty then of signal here that's specific to when MYC and PNUTS are both in close proximity. And the other thing I like about PLA is it's quantitative. So when, again, we assay for MYC alone or PNUTS alone, not so much signal, but together, lots of foci per cell. We also measure, remember MYC is a transcription factor. So we also measure MYC binding to different MYC target genes by classic chromatin immunoprecipitation, followed by qPCR. And you can see MYC chips very nicely to some known interactors shown in blue. And when we assay for PNUTS, we see that PNUTS is also chipping, if you will, binding to the same promoter regions of these MYC target genes. And we also did chip-re-chip. So if we pull down just where MYC is bound, we can also see that PNUTS is also similarly bound. So that gave us confidence that, in fact, the MYC was interacting with PNUTS, PP1 within cells. Okay, well, what's this interaction doing within cells? So the other nice thing about working with an enzyme is there's inhibitors. And so we could use Calyculin A as a tool compound because it inhibits PP1. And when we inhibited PP1, what we saw was remarkable. We saw this hyper-phosphorylation of MYC, and this is a Western blot here I'm showing you, control versus Calyculin-treated cells. And here we're measuring the phosphorylation of MYC, just using one phospho antibody, but of course we've used many. And so MYC is hyperphosphorylated when the phosphatase is inhibited. That makes sense. And MYC is degraded. That was a surprise. That was a pleasant surprise. And when we block the degradation using something like MG132, which is proteasome inhibitor, you can see basically, again, that hyperphosphorylation, but now it's augmented if we prevent the degradation. You can really see that hyperphosphorylation of MYC. And this regulation is not occurring at the level of RNA. When we measure cells in response to Calyculin A for the mRNA of MYC, if anything, we get a slight upregulation. That's basically, I think, a compensation as MYC is being degraded, right? You're getting this feedback response and so an upregulation of transcription. Okay, we did this not only with the tool compounds such as Calyculin A, we also validated using RNAi. Again, this is published, and we got the same sort of results. It complemented the pharmacological inhibition approach. We also tested numerous phosphatases, and we could see that really only inhibiting PP1 was leading to this hyperphosphorylation and degradation. We didn't see anything like that when we tested with PP2A, for example, another phosphatase. And we could also rescue this hyperphosphorylation if we put in exogenous PP1. So we know that PP1 is actually functional, this phosphatase on MYC. Okay, so what we also assayed was, well, what happens in response to Calyculin RNAi? And we saw that with ChIP experiments, this hyperphosphorylation and degradation obviously led to MYC no longer being bound to DNA. Well, we needed to validate that by an independent approach. And so we mapped using phospho-specific, basically phospho-enrichment mass spec, and mapped the sites of phosphorylation in response to Calyculin A. And we identified 12 such sites. Well, that was terrific because then we could make a mutant and convert those phosphosites to alanine, and that would prevent the phosphorylation. Or we can convert those phosphosites, serine and threonine, to aspartic acid, which is a phosphomimetic. And when we did that, we could see that here, I'm just showing chromatin immunoprecipitation, for example. You can see a number of different MYC target genes and a control. MYC is bound, as shown by ChIP, and the 12A mutant is bound. But if we had a phosphomimetic, just like with Calyculin treatment, when MYC is hyperphosphorylated, MYC can no longer bind to chromatin. And similarly with this these mutants, we were also able to validate that MYC degradation was also associated with the hyperphosphorylation. When we tested the 12A mutant, for example, and did not get the hyperphosphorylation in response to Calyculin A, it changed half-life of MYC from 30 minutes to 60. Okay, so basically that sets us up with a model that PP1 MYC interact. If we inhibit PP1, as we've shown you, we get MYC hyperphosphorylation and then degradation. So that's pretty good. I mean, that's one way to get rid of MYC, to drug MYC, to inhibit MYC, just degrade it. That's the good news. The tough news is that if we just inhibit PP1, that's not going to work, because PP1 has numerous substrates, and it would be cytotoxic to normal cells. We wouldn't get the specific inhibition of MYC and PP1. We would simply be destroying all PP1 activity. And lots of people have tried to drug phosphatases, and it's been shown that they're just generally cytotoxic. So instead, we need to actually not inhibit PP1, but instead inhibit MYC PNUTS interaction. And we anticipate if we just inhibit this interaction, that too would then drive the hyperphosphorylation and degradation of MYC. Well, that's where our first paper kind of left off. And the new sort of part of this story, we're just about to submit. And what we had to do is determine whether the interaction was direct, because only direct interactors can really be inhibited. And so that was also a criterion, go, no-go decision. And I'm talking about it so you can guess that the two interact directly. So here's MYC, and here we show at the basic helix-loop helix leucine zipper region, this is where MYC and MAX interact. And people have tried to drug MYC and MAX, of course, but after 3 decades of work, there is no inhibitor to MYC and MAX that can be used in the clinic. And in hindsight, I think that's not surprising, because many transcription factors have the BHLH leucine zipper moieties, and so there's nothing really unique there, and you get a lot of off-target effects. What we were interested in, by contrast, was really to identify interactors of the amino end of MYC, and particularly these MYC boxes. MYC box 0 to MYC box, there's 6 MYC boxes. They are unique to the MYC family and conserved between them. And so again, if there's going to be any interactors that are going to be unique to MYC and important for MYC activity, perhaps it's those that would bind through these MYC boxes. And in fact, we identified that PNUTS did interact with MYC through MYC box 0. This was a great day in the lab, because MYC box 0 is highly conserved between c-MYC, L-MYC, and N-MYC, which all drive oncogenic cancers. And we had just shown, in fact, MYC box 0 had not been really identified or characterized very much over the years of MYC research. But we had just shown, in fact, that in terms of MYC's ability to drive tumorigenesis, this is 4 different mouse models that we tested this in. When we deleted MYC box 0, in fact, we really curtailed MYC's ability to drive cancer. MYC box 2 is also very important, and that was known. So that served as a positive control. But MYC box 0, that was brand new news. So now we had an interactor of this new MYC box that was, in fact, critical for MYC to be a potent oncogene. We also mapped the interaction domain of PNUTS, shown here, to the amino end, which we called the PNUTS amino terminal domain, or PAD. And we like to think of this as the landing pad, if you will, for MYC interaction. And in fact, with a lot of work, we solved the structure of PNUTS PAD domain, that's shown in purple and in stick figure, and how it interacts with the MYC MB0. And I have to say, collaboration is the key to all successful science. I cannot be a structural biology expert as well. And so my colleagues, Cheryl Aerosmith, Maria Sunerhagen, are structural biologists. Bjorn Wallner is modeling protein-protein interactions, and David Andrews actually studies protein-protein interaction from a biochemical perspective. And it's with a team effort that we've really been able to show here in purple, this is a space-filling model of peanuts, and where MYC in yellow interacts. And you can see it forms this sort of a pocket, if you will, within peanuts, where MYC can actually take on the shape of peanuts because it's an intrinsically disordered protein, and it does so quite remarkably. So what we've learned from a biological perspective is that MYC, again, Maria's work has studied MYC from the structural perspective. In fact, she came on sabbatical to Toronto in 2008 to start working on MYC with Cheryl and I. And so we have learned over the years that in the amino terminus of MYC, there's an intramolecular interaction. And what we've been able to show is when MB0 lands on the landing pad of peanuts, it basically, these intramolecular interactions are relaxed, and that enables PP1 to really dephosphorylate a number of phosphosites located basically just C-terminus to MYC box 1. But I promised you that we'd be able to drug MYC if we learned something about MYC protein interactions. And so in addition to the biology, we've also pursued the idea of maybe targeting this MYC peanuts interaction. And for that, we developed what's called a fluorescence polarization assay. It's used in the drug development world. And this is, again, in collaboration with David Andrews. So we could develop basically an in vitro interaction assay and show that we could compete it out. So we had all the specificity required, and then use this in a high throughput manner to actually screen a number of compounds. And we just took a very small library of compounds, about 2,000 compounds, just to provide proof of concept that basically we could use this assay to basically evaluate which compounds might disrupt this MYC peanuts interaction in vitro. And we identified a number, and we've been able to validate those in vitro. None of them are good enough, if you will, based on my medicinal chemists that for further development. But it does provide proof of concept that we do have an assay. We can identify inhibitors of MYC peanuts interaction. And so we think we might be on the road to actually maybe developing an inhibitor to MYC with high specificity, sensitivity, and durability by interrupting the MYC peanuts. And I say this because specificity, well, MYC only, I think, takes on this shape with its interaction with peanuts. We have other MB0 interactors, and so we're testing that hypothesis. Sensitivity, because MYC, if the E3 ligase or degradation machinery is disrupted in a cancer, that's okay, because MYC actually is degraded by at least a dozen mechanisms. So inhibition of any one of those is likely not to confer resistance to this drug, this inhibitor of MYC peanuts interaction. And durability as well, because it's not dependent on any one phosphorylation site. We get a hyper-phosphorylation of at least 12 sites. So we're not going to get mutation of any one amino acid that then confers resistance. So I have to say, I'm pretty excited by all this. For me, it's like a lifelong journey to actually develop a MYC inhibitor. And so our hypothesis is, again, that if we can disrupt MYC peanuts interaction, we can drive this regulatory axis to lead to MYC degradation. And so stay tuned. We're now launching some screening campaigns, as they're called, of much larger drug libraries to see if we can indeed identify a drug-like molecule that can actually go forward to full drug development and actually impact patient care. Go big or go home, basically, was my motto in terms of choosing to study MYC, and I hope we get there. Fingers crossed. I'd like to thank the people in the lab that did the work. Diana Rasetka, she's our mass spec expert, really helped establish all of BioID in the lab, working with Brian Rout. And I have to say, she just had a baby boy. And Yang Wei, he's a structural biologist who really did the bulk of the work on PNUTS, along with our collaborators, in terms of all the nuclear magnetic resonance that needed to be conducted in order to understand the nature of the interaction. And Connie, she's been validating the interaction in cells and actually evaluating point mutants of MYC and PNUTS to show that, in fact, we do understand the contact points that are important, because mutating any of those sites, either in vitro or in cells, disrupts the interaction. And again, my collaborators, Brian for all the mass spec, Cheryl and Maria, all the NMR, and David, all the protein-protein interaction work, particularly in vitro. And of course, you have to thank the funding agencies. If you want to get anything done, you have to put money behind it. And so I'm very grateful that they have provided us with the resources to allow this work to go forward. Happy to take any questions. Thank you.
Thank you very much, Linda. That was an outstanding presentation, which certainly has given us an insight into how long it takes to go from studying a protein to taking it all the way through translation, how persistence pays off. So there are a couple of questions, but being the moderator, I can jump in and ask mine right away. So you've clearly indicated that going this route will avoid resistance, which is great, and you've identified one target. Do you anticipate that using more than one target will be effective? So you've identified PNUTS and you're going through the process of screening libraries for others. Combination therapy here?
Oh yeah, so my colleague Bill Tansey, he identified WDR5, for example, as interacting with MYC-box4, and he is drugging that now. And we're talking all the time, and we're excited. I mean, we're excited to have an arsenal of inhibitors to MYC. All these years not having a single one, if we can develop, I want hundreds, and then we can learn whether we need to use them in combination with classic chemotherapy or a drug cocktail of MYC inhibitors. This is all in the next, I think, near future. Certainly, I think within the next decade, there's going to be a number of, largely the field has been broken open by these the interactome, and so it's quite exciting to see everybody. And people always say, "Oh, aren't you scared people are going to run away with it? It was so nice of you to share all your data." And I'm like, I'm so excited. I can't do all this work. I want everybody to do it. So it's really, really gratifying to watch and learn how things are moving forward. So we'll see. Maybe it's PNUTS. Maybe it's one of our other. We're also working on TF2F as an MB0 interactor. And we also have identified TBP. So MYC is a transcription factor interacting with a lot of general transcription machinery. So we're also taking that approach. There's a number of different strategies.
Other questions. So why use libraries if you know you've got this model of how everything interacts? Why not just do de novo medicinal chemistry and come up with some de novo ones? Yeah,
you're bang on. So Wednesday morning, I was talking with Cyclica and other companies that do a lot of modeling. So now that we have the structure, without the structure, we couldn't go this direction. But now that we understand precisely, I mean, at a molecular level, how these proteins interact, now we can start to model which drugs are most likely to be useful inhibitors of this protein-protein interaction. So we'll enrich for those and then screen. Absolutely.
So somebody who you know well, Bahati in Kenya, has asked a question. Just curious to know whether MYC dysregulation is differentially affected in hormone-driven tumors and whether the stage of the cancer inhibits the possible druggability of MYC.
That's a really good question, Bahati. I'm not surprised you've asked such a good question. Nice to hear from you. So MYC was a major player in all cancers, as I mentioned, pediatric as well as adult. Breast and prostate are particularly sensitive to, are particularly driven by MYC, and those that are most aggressive. So triple negative breast cancer, for example, where terrible disease, no treatment options, affects young women. It's we need an inhibitor there. It's a major unmet need, and that's basically where we work primarily, is in breast cancer in triple negative. Also in prostate cancer, the most aggressive prostate cancers have a deregulation of c-MYC or n-MYC, even l-MYC, in fact. And so there again, a MYC inhibitor would have, I think, a significant contribution. Advanced cancers. MYC is often deregulated in advanced cancers. Once we have the tool compounds, even tool compounds is what I'm after, so we can ask those very important questions, because we haven't been able to until we have some tools in hand, and it'll be one of the first things we ask, of course. Thank you.
Okay. Pam, have you got a question, or should I go from the Q&A?
Well, why don't we, since we have Linda here, I think we'll follow a few minutes behind, but let's cover all of it. If you can go to Q&A, and I think Ashton has questions, and I'll ask a really quick one at the end. Really great talk, by the way.
Okay, I hope I didn't go too fast. Sorry about that if I did.
A quick question we got here is, how does MYC control the balance between self-renewal and differentiation?
Yeah, great question. In fact, MYC drives stemness and self-renewal, and that's another indicator of why it's such a potent oncogene. And differentiation, numerous different model systems. If MYC is deregulated, differentiation just cannot take place, because you basically have to exit the cell cycle in order to differentiate. And in fact, as I said, with the tool compounds, with the switchable alleles, in some cancers, when MYC is turned off, the tumor does differentiate, and that's basically one way to curtail the cancer. So great question.
Over to you, Payam.
Ashton, go for it. You go first.
Okay. Dr. Penn has a fantastic presentation. Very much appreciate it. I have a couple of questions, more than that, but last two. The first being...
Love the enthusiasm.
You spoke about a lot of the strategies for targeting the protein-protein interactions. Can you talk about the challenges between targeting the MYC-MAX interaction with the DNA? I know it's the basic helix region it binds to, but just the basic, I think, binds to the actual DNA. Are there drugs or strategies for that particular regions of it?
This is a smart crowd. That is a great question again. In fact, we tried that route. We developed something we call affectionately, what was it called? ME47 or MAX47. And basically we made a protein, it was a fusion protein of the basic region of MAX with the helix loop helix region of E47, another BHLH leucine zipper protein, so that it would function again as a dominant negative. So it could interact with E boxes with higher affinity than MYC and MAX. And it worked theoretically. And in both in tissue culture, we could show it displaced MYC and MAX in terms of chromatin immunoprecipitation assays, binding to DNA, and also was great in xenograft experiments where we induced the ME47. So very similar to OMO-MYC. In fact, I should use my own examples for those kinds of proof of concept. But putting it into a pill became the rate limiting step. So again, it was a small protein. It was a 57 amino acid protein. We tried to use nanotechnology, nano delivery, really complex, because it had to get to the cells, and it had to get to the cancer, it had to get to inside the cancer cells, inside the nucleus. Yeah, right. We'll probably revisit it when nano delivery comes to speed. Sure.
And actually, that was going to be a follow-up question to piggyback on what Eleanor talked about, the de novo approach to making a molecule. You mentioned cyclica. So in terms of making a biologic, a protein biologic that has maybe a higher affinity than MAX for MYC, but that cannot bind DNA. But you already mentioned you've tried that in terms of a small peptide, right?
Yeah, people have tried numerous. I showed you OMO-MYC. Other people tried other approaches. All great in theory. Just great tool compounds, just not drug material. You know, taking it through to full drug development. No, I mean one person. So Laura Susek, she's trying to take OMO-MYC forward. She thinks that she can. She says she's going to try a clinical trial, start a clinical trial next year for, I think it was esophageal cancer, where maybe drug delivery isn't as challenging. So we'll stay tuned. I hope it works. Yeah.
Fantastic. Can I ask just one more quick question, Payam?
Of course, ask as many questions. I'll skip my question back.
Just in terms of when you establish your whole MYC interactome with your BioID strategy, is there still when it folds, there's still no conserved structure that's druggable after you have all that whole interactive information? Or it's still like it depends on the class that it's interacting with?
Yeah, good question. Early days. We've only that was published December 2018, so it's still only a year and a bit with COVID. So we really haven't had too much time yet. We're basically validating the interactors and evaluating exactly that. So, for example, the 3 hits that we got in our little screen for MYC PNUTS, we have another interactor for MB0 TF2F. Those small molecules don't inhibit MB0 TF2F interaction. So we're hopeful, but it's early days. But we have those.
Okay, thank you so much.
That we'll be able to answer those questions.
Fantastic. Thank you so much, Dr. Penn.
It's been fun. Thank you. Thank you so much.
Thank you so much for joining us. Eleanor, how about in the interest of time, should we move ahead? And maybe if there are other questions, if Dr. Penn would like to stick around, people can ask in the chat section.
Q&A, absolutely. Linda, if you don't mind taking a look at the Q&A and answering some other questions. But thank you very much for a very exciting and hopeful strategy that will be effective.